On the behalf of KTHM College Nasik and Department of Chemistry of this college, I am Dr. R.K.Pawar welcomes you in this M.Sc. Secondary Inorganic Chemistry class. Dear student, in this lecture we are going to discuss the metalloenzyme of copper, that is galactose oxidase. Galactose oxidase is the family of oxidoreductase enzyme. In galactose oxidase, as we see that the copper is an active site. The speciality of this enzyme, it is a free radical enzyme where copper is an active site. The name of galactose oxidase, it catalyzes the oxidation of D-glucose in species of fungi. So let's discuss the structure, function and reaction of galactose oxidase. Structure. As we know that the copper is an active site. In structure first, as we know that the galactose oxidase is a type 2 copper protein, and in that the active site is a copper. So here the galactose oxidase is type 2 copper protein. It is in that the copper is an active site. In that copper is an active site. Copper is attached to, copper is bound, or we can say that the active site copper is bound to two tyrosinase molecule. In that, preferably tyrosine 495 and tyrosinase 272 residues are bound to the active site of copper here. So the two residues, that is tyrosinase 495 and tyrosinase 272 residues are bind to copper or the active site. Third point is that here the third and fourth positions are generally bind, means the two histidines are bind to the copper. So here the third and fourth position is coordinated with two histidine simultaneously bind to nitrogen, two histidine residues. And as we see in structure that here the fifth position is actually occupied by the solvent molecule, that is usually water is used as a solvent, and which is a very labile position here. So in fourth point we can say that the fifth position is occupied by water or solvent molecule. Generally water is used as a solvent here. So The fifth position is occupied by the water molecule and it is a labile one. It is labile. Means it can replace and can replace by now in substrate molecule. Here now if you see the geometry of this respective metalloenzyme of galactose oxidase, generally square planar or square based pyramidal geometries are adopted. So the geometry of galactose oxidase square planar or square based pyramidal. The point is that here if you see the tyrosinase 495 is situated at axially, whereas the other ligands are lie in the plane. So in galactose oxidase, tyrosine 495 residues are at axial position and others ligand are at plane. Now the next, that is in the fully oxidized form of galactose oxidase, the free radical couples to copper center antiferromagnetically and it is supported by the X-ray data and supported by EPR studies. So this is the general structure of the galactose oxidase. Now let's discuss what are the reactions of galactose oxidase. As we know that in the oxidation of galactose is nothing but the galactose oxidase. So the primary function of this galactose oxidase is the oxidation of galactose. But in yeast, the galactose oxidase play a following reaction. D-galactose plus O2, it gives diols plus H2O. it can oxidize the galactose to hexadiols. So this is the main product of that reaction. Now instead of that, next one is the reaction is essentially the oxidation of primary alcohols using dioxygen to form the corresponding aldehyde and hydrogen. So likewise galactose oxidase also oxidize the primary alcohols. So now the next reaction is, as we know that the galactose oxidase can oxidizes primary alcohols other than galactose. So the third important reaction, that is galactose oxidase or catalase dihydrogen hydroxyacetone 3 times faster than it does to galactose. So this is the reduced selectivity of galactose oxidase. So it cannot oxidize secondary alcohol Now the next reaction is that the 2-electron oxidation is achieved by the double redoxide copper-seeking metal center and free radical, each capable of accepting one electron from the substrate. So this is a general reaction. So in this reaction, the double redox method has 3 oxidation steps. So in galactose oxidase, the copper is in an active site. It will shuttles between the oxidized form and fully oxidized form to fully reduced form, and there is one middle form, that is a semi-oxidized form, which is an inactive form. Let's see the catalytic cycle of galactose oxidase. The copper shuttles between fully oxidized to reduce, fully oxidized form of copper to fully reduced form of copper. So here, if we see the reaction, it's as like this is a fully reduced form of copper, and this is a fully oxidized form. We can write here. And this is a semi-oxidized form, which is an inactive form. These are the reactions which are catalyzed by the galactose oxidase. Now let's discuss what are the applications or the functions of the galactose oxidase. So let's discuss the functions of galactose oxidase. In bioanalysis, now let's discuss the functions of the applications. Bioanalysis. Now, biomolecules. Such as, we know that the galactose oxidase can oxidize the galactose. Instead of that, it also play a very important function in biomolecules. Biomolecules such as galactose can be quantified by using oxygen, that is O2 detection. Galactose can be quantified by the oxygen detection method. If suppose, since we can take an example, consumption of oxygen corresponds to one equivalent primary hydroxy group oxidase then that here biomolecules such as galactose oxidase can be quantified using oxygen detection method, since one equivalent consumption of oxygen corresponds to one equivalent primary hydroxy group oxidase. Now the second application is that galactose oxidase utilized as a biocatalyst in synthesis of aldehyde and carboxylic acid. Now the next application is that the formation of H2O2, that is hydrogen peroxide, during substrate oxidation can also be used for colorimetric detection of galactose using dyes and these dyes are generally oxidized by which are oxidized by hydrogen peroxide. This is the important function of the galactose oxidase. Now the next function is that in organic synthesis also galactose oxidase play a very important function. So in organic synthesis galactose oxidase used as a biocatalyst in synthesis of aldehyde and carboxylic acid primary alcohols alcohols. So these are the general applications of galactose oxidase. Now we will stop here. In next lecture we are going to discuss the next metal enzyme of copper, that is SOD, superoxide dismutase. Thank you.